Okay, so I wanted to tell you about lab test 1. Lab test 1 is going to be what we call practical, which means it's set up across the whole room with stations, and each person will stand at a station. The station will have a card, and the card will have questions with multiple choice answers. Most stations will have some kind of visual with it, like maybe you're supposed to answer parts of the microscope, or you're supposed to measure the length of a bone. Okay, there's some kind of visual at each station to show your knowledge or your skill. And again, it is multiple choice. It's 50 questions. 50 questions. And so this lab review is recorded. I'm recording right now as I'm speaking, and whatever you see up there is also being recorded. And then when we finish class, I post it on YouTube. So if you want to go back and review any particular portion of the lab review tonight or tomorrow night, you can by accessing it on YouTube. You search for my name. So if you're interested, now it's totally optional, but if there's any part while you're reviewing you don't remember what I said, or you didn't write it down, or you just want to listen to the whole darn thing again, you just search for my name and then pick our class, because a lot of classes will come up. So you just pick our class. So if I have a question about that.
It's going to be listed by lab time?
By lab time.
Okay.
8:35 lab, fall 2016.
Okay.
That's a good question. Okay, so lab number 1 was the scientific method. And we did an activity one day using earthworms. I think you had a handout on that had the steps of the scientific method. Does anybody remember the 4 steps of the scientific method?
Observation.
Mm-hmm.
Conclusion.
Good. Okay, so observation, hypothesis, some kind of testing, which would mean experimentation, and then based on your data, you come up with a conclusion. And I want to remind you, your conclusion is that your data support your hypothesis or your data do not support your hypothesis. And again, this is on a handout that you had for that activity for that day. Okay, if given an experimental variable, be able to determine the experimental group, control group, experimental variable, and data. I didn't read that correctly. Yeah, well. So what I'm going to do is describe an experiment, and you have to pick out what the experimental group is, the control group, experimental variable, and data. So I'm just going to do something for pretend, okay? I want to go to, in your lab manual, would you turn to page 30? We did an experiment on page 30 in your lab manual on the iodine test for starch. I'm going to start with the easy question. Remember, this is the iodine test for starch. We have 2 control test tubes. One's a positive control, one's a negative control. What was the positive control for this experiment?
Starch.
Test tube number 2, the starch. What's the negative control for this experiment? Water. Test tube number 1, the distilled water. Okay, what were the experimental groups? Test tubes 3, 4, and 5, where we didn't know what the answer was going to be, and so we had to test them to figure out the answer. So the experimental groups were test tube 3, 4, and 5, and the controls were numbers 1 and 2. Okay, here's the third bullet up here. What was the experimental variable? The contents of the tube. Look how that changed tube to tube to tube to tube. Okay, now we put the same volumes every time, and you put the same amount of iodine in every time, but what made test tube 1 different from test tube 2 was the contents, correct? So experimental variable is what changes group to group to group to group. So it was the contents of the tube was our experimental variable on that experiment. Okay, and then lastly, what was the data that we collected? Oh, those are our conclusions. What is the data we collected?
The colors of
Color. We collected color data. You see how your data is the color. That's the data. That's just using one experiment as an example. And it was an experiment we did. But you can see we kept doing these kinds of experiments over and over again, and every time you can go back and pick out which tubes were your controls, which tubes were your experimental groups, what was your experimental variable, and what data did you collect. Is everybody okay with that, as using this as an example? Okay, great. Okay, you have to be able to write a hypothesis. And I recall we practiced that. I want to remind you, most of the time a hypothesis is written as an if, then, because sentence. Like, if earthworms are placed in a dry environment, then they will move towards moisture because earthworms dry out without moisture. That's a hypothesis. If, then, and because. Remember that a hypothesis is an educated guess or a tentative explanation. You're trying to explain what you think you're going to see happen during the experiment. Okay, and then based on your data, be able to reach a conclusion. Please don't forget, conclusions are based on your data from your experiment, and your conclusion either would be the data support my hypothesis or the data do not support my hypothesis. Those are the only two possible conclusions you can reach from an experiment. So everybody okay on exercise number 1, scientific method? Anybody need anything repeated on exercise 1, scientific method?
For the control group for the starch experiment, you said it was the water.
That was our negative control.
And the positive control was the starch.
Correct. We use negative controls more often than positive controls, but sometimes we are able to have both. Anybody else? Okay, great. I want to move to exercise 2, metric measurement. Now, this actually takes us back to our very first day together. And on our very first day together, we started using some metric prefixes, like milli and centi. Those are the more common ones that we use. So let me talk to you about those. Here I'm using them. And I want us to be able to fill out these blanks. Everybody look up at the screen. So we said the standard unit of measurement for length in the metric system is the meter. And then we said that we divide the meter up into centimeters. So you better remember how many centimeters there are in a meter.
100.
That's correct. Okay, then we took centimeters and divided them up into even smaller units called millimeters. How many millimeters are in a centimeter?
100. This is 1,000 millimeters in a meter, is there not?
There is. It's 10,
Oh, yeah.
of course. And then, as Rick said, there's 1,000 millimeters in 1 meter, because 100 times 10 is definitely 1,000. Is everybody okay on what I have on the screen so far? Okay, great. Now, once we started developing good microscopes, it turned out that millimeters were too big. And we actually proved that to ourselves one day. We put a ruler on the microscope and measured the field of view diameter with a ruler. And I remember by the time we got to high power, the field of view diameter was less than a millimeter. So microscopists came up with some more units. And the first unit I want to tell you about is the one that has the fancy U up here. Somebody see it up here? they wanted to have a unit called a micrometer, M-I-C-R-O, micrometer, but they couldn't use M-M for micrometer. How come? It's already being used. So they used the Greek symbol for micro, which is this fancy looking U. That's the symbol for micro. And so what they did is they took 1 millimeter and they divided it into 1,000 equal units. And so 1/1000th of a millimeter is a micrometer. Also pronounced micrometer, if you ever hear it pronounced that way, micrometer. Most of the pictures that you will see in your book have the micrometer or micrometer symbol on them as a unit of measurement. It's a very common unit of measurement for pictures that are taken from microscopes. Okay, then in the 1940s, they developed the electron microscope, and the micrometer or micrometer ended up being too big to use with electron microscopes because the magnification on electron microscopes is so high that they needed another unit of measurement. So they came up with this one, NM. Does anybody know what the N stands for? Nano. Yeah. So they took 1 micrometer and divided it into 1,000 equal units, and that's what we call a nanometer. So if you think micro means small, because it does, then nanometer means really, really small. And so can you do the math up here for our last blank? How many nanometers would it take to make a meter? I'm sorry, that should be a millimeter. It's an error. It is 1,000 times 1,000. The question is, what is 1,000 times? It is a million. Yeah, it's tough. Okay, guys, so I want you to real quickly find a ruler on your tabletop. Now, make sure you're on the metric side, and I want you to find a millimeter. Millimeters are the little black lines, right? Okay, can you imagine that divided into 1 million little units?
I feel like it'd just be a black line.
It would be, yeah, it's so very small. Okay, so a nanometer is very, very, very small. Okay, great. All right, so number 2 is to make some conversions. Using the information we just wrote down for number 1, we want to convert 23 centimeters into millimeters. Anybody?
Centimeters into millimeters?
Yeah, we're going to go 23 centimeters into how many millimeters?
230?
230. Because it takes 10 millimeters to make a centimeter, and if you have 23 centimeters, then you have 230 millimeters. Okay. Okay, now we're going to convert 368 millimeters into micrometers.
368,000.
Mm-hmm. Because it takes 1,000 micrometers to make a millimeter, we have 368 millimeters, so we have 368,000 micrometers. Okay, then lastly, we're going to take 6 micrometers and convert them to nanometers. Yeah, that one's easy, 6,000. Everybody okay on that one? The conversions? So we used the values we wrote down for number 1 to make the conversions on number 2. Okay, great. All right, looking at the same ruler. You should have a little plastic ruler on your table. Make sure you're looking at the metric side. See if you can fill out these 2 blanks. If you have a lab partner, they can help you. Or maybe they need your help.
Are we looking at how many centimeters? How many?
On the ruler. Yep. How many centimeters, how many millimeters are on the ruler that's on your tabletop?
15 centimeters.
Everybody in agreement we have 15 centimeters? Yes? So how many millimeters would that be?
150.
Everybody okay on that one? We all in agreement? Yes. Okay, great. Okay, here's some more easy questions. What are the freezing and boiling points of water? Let's start with freezing. And guys, it has to be in Celsius. Why? That's what we use in science. So in Celsius, what's the freezing point of water?
0.
And boiling?
100.
Okay, number 5 is about skills. It says, be able to measure, weigh, and determine the volume of an object using appropriate metric units. Okay, so let me just start with this one. Let's say I gave you this seashell on the test, and I asked you to do its mass. What piece of equipment would you use? The scale, the triple beam balance or the scale. I just want to remind you when you're using the triple beam balance, also known as the scale, that when you move the weights, make sure the weights fall into the notches. The notches. It's the only way you can mess this up, is if you don't put the weights down into the notches on the balance. Okay, now what if I want you to do the volume of this shell? What would you use? Graduated cylinder. And some water, right? Need some water? Question, do you put the water into the graduated cylinder before or after the shell?
Before the shell.
Thank you, before the shell. And do you have to know how much water you put in?
Yes.
Yes. Why do you have to know? Because you have to do subtraction. You have to know how much is in, then you put in the shell, then you see how much it is now, and you do subtraction. So water goes in first. Okay, you guys crack me up. You put the shell in, then you pour water on it, and you stare at it. You're like, okay, so water first, then the shell, then look at the difference. But it's okay on that. Okay, so what about this? I want to do the mass of this box of tissues. What do I use? The mass. Why is this hard? The scale. I want to use the scale. Now I want to do the volume. What do I use? Renee's right. Length times width times height. Now, what unit in the metric system should you use? Centimeters. Yeah, I want you to use centimeters. Length times width times height in centimeters. Now, you need a calculator to do that. Can you use your cell phone? No. So bring a calculator. I have a few that you can borrow. I have a few, but I need you to bring a calculator, a simple calculator. This calculator, the most important thing, if you don't have a plain old calculator and you're going to go buy one at Walmart or whatever, needs a square root button. Later in the semester, we will need to do square root. That's all. You do not need a scientific calculator. I know it's a science class, but you don't need it for this class. So you need to bring a calculator. Louis? No. Length times width times height is for the volume. Right? Length times width times height is volume. Again, what piece of equipment do you use to do mass? Scale. Okay. Okay, number 6, what is a meniscus?
That little dip in the measurement of a graduated cylinder.
Excellent. It's the little dip in the graduated cylinder after you put water into the graduated cylinder. And then I have this little, and you can tell I have an excellent drawing up here also. And then you can see I have pink liquid in my graduated cylinder. How do you properly read a graduated cylinder? The bottom. Thank you, Dominique. The bottom of the meniscus. The bottom of the meniscus. Now, I have to warn you that on my graduated cylinder questions, all of my values are very close. Like, I'm making this up, 30, 31, 32, 33. I put the liquid in and you have to measure, so you actually have to know what you're doing, okay? Like, it's not like I put in 30 milliliters and give you 50 as a choice. Like, duh, I'm not doing that. Okay, they're all real close. So you got to know what you're doing. Make sure you get down eye level to the meniscus and keep the graduated cylinder on the tabletop. Why? Why do you keep it on the tabletop? I need a level surface. Keep it level. So bend down and get your eye level to the meniscus. Anybody have a question about exercise 2, the part called metric measurement? Okay, exercise 2, microscopy. Would you join me on page 14 in your lab manual? Page 14.
What page again? Sorry.
14, page 14. Okay, guys, so in this lab, we have 2 kinds of microscopes that we've used so far. We've used the compound light microscope, which is the one that's right behind Tyler. Got a whole cabinet full of them. Remember those? They have the red handles. And then on a couple days, we used what's called a stereoscope. This was a scope that you used to look at your earthworm in a little more detail. And then one day we just used them to look at a variety of things like butterfly wings or flowers, just a variety of things. Okay. And so, again, let's just pretend that you're at the beach, you collect some seashells, you bring the seashells back to the lab, and you want to look at the seashell in more detail. You want to see more detail on it. What kind of light microscope would you use?
Stereoscope.
The stereoscope. Yeah, because you can't make a slide out of this, right? The compound light microscope, you have to be able to make a thin preparation of whatever it is you want to look at, because light has to be able to go through it for a compound light microscope. But on a stereoscope, the light just bounces off. You remember a stereoscope is like a really nice magnifying glass. It uses reflected light, not transmitted light. Okay, let's say you were at the beach and you collected some seawater that had algae in it. You brought the water back to the lab and you wanted to see if there were any microorganisms in the seawater. What type of microscope would you use?
Compound light microscope.
The compound light microscope. And what's the name of the slide preparation you would prepare? A wet mount. You guys remember that one? Wet mount? Wet mount. Okay, good. A wet mount. W-E-T, wet. Is what we would use. Our preparation of the slide would be called a wet mount.
And the type of microscope is
Compound light.
compound light.
Okay, on page 15 is just a real quick comparison of compound light microscopes and transmission electron microscopes. The reason they're comparing those two is because both of them require energy to go all the way through the specimen for you to see it. Compound light microscopes, that's why you have to prepare your wet mount thin so light can go all the way through, transmit through. And again, a transmission electron microscope, the specimen will have to be super thin, super thin. But we wouldn't memorize anything on there, but I do want to point out number 4. Let me take a look at number 4. The very best compound light microscopes can magnify to about 2000x. Look at the very best transmission electron microscopes. What is that number? 1,000,000. 1,000,000. So I think you can see the advantage to the development, invention, and then development of the transmission electron microscope. It opened up a whole, just like the compound light microscope opened up a whole new world to us in the 1600s, the transmission electron microscope did the same in the 1900s. Okay, great. Then you see a difference of cost there too. That's why we don't own one, but that's not the point right now. So let me tell you some of the things that we've been able to see since the development of the electron microscope. So we already thought they existed. There are these particles that cause disease called viruses. Now we've been able to see them, okay, because they're so small you can't see them with the compound light microscope, but we can not only see them, we can see them in detail now with the electron microscope. You guys heard of viruses?
Yes.
Okay, great. And then with the transmission electron microscope, we've actually been able to see large atoms. Now, when I mean large, I do mean with high molecular weights like uranium, which has a molecular weight of like 235. Not hydrogen. You guys remember hydrogen has a weight of 1? You haven't seen it yet, okay? But big atoms, we've been able to see it with the electron microscope. So again, it's just opening up new insights into our natural world. But okay, on number 2, just a quick comparison of light microscopes versus electron microscopes. We already talked about number 3. Know how to use a dissecting or stereo microscope and what can you view with it. Remember, stereoscopes are great for magnification of any item you want to see in more detail. Again, it's just kind of like a great magnifying glass. Okay, number 4, I want you to star because I'm going to ask you several questions from number 4. For number 4, would you join me on page 18 and 19 in your laboratory manual? On page 18 and 19, that day you should have filled out page 19 by labeling parts of the compound light microscope. And then we went through each part and talked about how to use each part. So I want you to go back and review the parts of the compound light microscope and then what each part does. And of course, what each part does is called function. So the functions of the part, and then what's the name of the part. That was page 18 and 19. Make sure that's starred on your review sheet because you're going to have several questions over question number 4 about the parts of the compound light microscope. Okay, now you guys may remember on a compound light microscope there's a 4x objective, a 10x objective, and a 40x objective. Just to review that, would you turn to page 21? There's a table there that shows our objectives. Our 4x objective is known as scanning, our 10x objective is known as low power, and our 40x objective is known as high power. We didn't have an oil immersion. Of those 3 objectives, which one are you supposed to start with? Scanning. Does anybody remember why?
You get a much wider field of view.
It gives you the biggest field of view. And I think we actually even measured it as a class. I think it came out to like 5 millimeters was our field of view. And so Rick's point is that you can see the most of the slide with the 4x scanning, so you can scan around and find what you want on scanning. Okay, then you find what you want, and then what do?
Switch to low power.
Not yet.
Center it.
Center it. Got to center it. And then you go to low power. What do you do on low power?
Center it again.
Focus and center it, and then you go to high power. Now, on high power, can you use the coarse and the fine adjustment?
Just the fine.
Just the fine. Yeah, when you get to high power, there is the requirement that you only use the fine adjustment. Okay, this is still on number 5. Be able to calculate total magnification. We did that on page 21 in table 2.3. What's the basic calculation for total magnification? Y times 10.
Well, the ocular lens is 10, objective lens is 4, so 10 times 4 is 40.
Okay, so it's ocular times objective equals the total magnification. It's the basic calculation. The ocular times the objective. Do I need to memorize all of the magnifications?
No.
No. Why?
Because
They're labeled on the microscope.
Thank you very much. Zach said because they're already inscribed on the microscope. The microscope's going to be sitting right in front of you on the test. How do you know on a microscope what the magnifications are? Because there's just bunches of numbers. How do you know which ones are the magnifications? That's right. Xenia said the ones with the X on them. Yeah, they have an X with the number, they're a magnification. But it's okay on doing total magnification.
Are you talking about like the side?
Okay, so the ocular is the part at the top that you look through, and it'll have magnification with an X. And then the objective, so Daniela's saying make sure you look to see which objective is clicked into position. So if the 4x objective is clicked into position, you use 4. If the 10x is clicked into position, you use the 10. So look which objective is lined up that you would be looking through if you were using a scope. Correct, Daniela? That's your point? Yes. Okay. Okay, we already answered this question. Which objective gives the largest field of view? Scanning, also known as the 4x. Okay, page 20 is where I want you to look for the word inversion. Page 20. Excuse me, it's kind of in the middle of the page, page 20. What does that mean, inversion?
The image on the microscope is upside down and reversed.
Good. What letter of the alphabet did we use to study this?
E.
So if a letter E looked like this on a slide, under the microscope, oops, it would get bigger. You okay with that?
Yep.
And then the top became the bottom and the left became the right.
Last time you saw a label.
But okay, on inversion, how does inversion relate to microscope usage? So, like, why should you care? How does it affect anything in your life when you're using a microscope?
Well, when you're scanning, you've got to know that when you're moving to the left, that the image for the deal itself is moving to the right.
So all your movements are opposite.
Yeah.
So if you move your slide up, the image goes down. If you move your slide to the left, the image goes to the right. So all movement is opposite under the microscope. So better remember why microscopes invert images?
The mirrors.
Because of the mirrors. That's good. Okay, does anybody need anything repeated on exercise number 2, microscopy? Okay, super. For our next one, I want you to join me on page 40, 40. And on page 40, you constructed a useful table that will be on the test. Can you recall what is the chemical that's used to test for protein? Biuret. Good.
Is that how you pronounce it? Violet.
Mm-hmm. Yes, sir. Violet. And then what is the positive color?
Purple or dark.
What's the negative color?
Blue.
But okay, on protein, testing for protein. Okay, I need the chemical used to test for starch.
Iodine.
Positive color.
Dark color.
Negative color.
Amber.
But okay, on what we use to test for starch. What is used to test for sugar?
Benedict and 3 minutes of heat.
Positive colors.
Orange, red, yellow, or green.
Negative. It should seem familiar. How do you use brown paper to test for lipid? First, you got to put it in the microwave to evaporate the liquid. And then what do you look for? Translucent spot. If you see a translucent spot, you've got lipid. It's okay? Okay, now I want you to join me back. And just want to comment one more time. Page 40 is an important page to review. Page 28 and 29. Some of this stuff was on our lecture test last week. Like, what are the building blocks of carbohydrates? You know, what's the smallest unit or monomer of a carbohydrate? Glucose is an example. What's a more generic word for the basic building block of carbohydrates? Who said that? Good job, Naomi. Monosaccharides, which means one sugar. Mono means one, saccharide means sugar. And if you want to reference page 28 and 29 in your notes here for number 2, feel free to do so. You have real good artwork in your lab manual for this. Okay, building blocks of lipids, page 34. I think this was straight up a question on our test last week. Yeah, I think I said what's a lipid or what's a fat or oil made out of, and you had to pick, and there was a list of possibilities. So let's look, page 34. What is a fat or an oil made out of? It's pronounced glycerol and fatty acids, that's right. Right there on page 34. I hope you got it right on the test. Some of you did. Ariana, did you get it? No. Mariah, did you get it on right on the test? Okay, 'cause you're shaking your head like, yeah, got it. I'll get it right again. Ready? Okay, on this question. Super. Back it up a page to page 33. Now I need the building blocks of proteins.
Amino acids.
Everybody knows that. So amazing. Okay. Just stay on page 33. At the top of the page, you see one amino acid joining with another amino acid to make two amino acids linked together called a dipeptide, plus water. Does everybody see the chemical reaction at the top of page 33? How are these monomers, like amino acids, combined to produce polymers? Well, it's a kind of chemical reaction where one thing is hooked to the next thing to make something bigger plus water. What do we call that?
Hydrolysis.
Dehydration. And again, page 33 is a good page to reference, but it's repeated over and over in all the visuals in the lab. Okay. Staying with the same visual. Now let's take this dipeptide and water and break the dipeptide into two amino acids. So say we're taking something big and breaking it into two little pieces. What kind of chemical reaction is that? That's hydrolysis. And I think that was also a question on our test last week. Excuse me. Okay, I need a definition of a monosaccharide. What's mono mean?
One.
Saccharide? What's an example of a monosaccharide? One of our favorite ones we've been drinking more and more of is fructose. I just want to mention it because you might like it. I don't know. It's from corn syrup in your Coke, Sprite, Dr. Pepper, Mr. Pibb, A&W root beer, whatever. What's a disaccharide? It's an example. Okay, I'll give you a hint. There is an example on page 28. Maltose. Who told me that? Good job. It's Ariana? My personal favorite disaccharide is sucrose. You guys have table sugar at your house? Me too. That's sucrose. I kind of like it. What's a polysaccharide?
Many sugars.
We need an example. I'll give you a hint. Page 29. Starch. Anybody else? Okay, starch is not my personal favorite polysaccharide because starch breaks down into glucose, and glucose makes you fat. What is my favorite polysaccharide? It's what my mama calls fiber. I call it cellulose. How come I like cellulose? It helps move food through my food tube. Can you digest cellulose? No. So it makes you feel full. It helps with weight control. Ms. Steinke, before she walked in here, she had a fiber 1 bar. 9 grams of fiber per bar. I feel full. Drank a whole bottle of water to go with it. Just saying.
How much fiber do you feel is and should be in a proper diet?
Probably 36 to 40 grams per day.
Definitely don't get that.
Okay, well, you should be. Yeah. Fiber 1 bars. What is a common energy storage form of glucose in plants? Okay, I'll give you a hint. It is a polysaccharide. plants make it, and then they store this glucose in some plant parts like potatoes. It's starch. So they make glucose in photosynthesis, and then they make all that glucose into starch for long-term storage. Humans have figured out what plant parts store starch, and that's what we usually harvest. Like potatoes, corn, any grain is loaded with starch. You know, wheat is loaded with starch, rice is loaded with starch, rye, barley, any of your grains are loaded with starch. Can you digest starch? Yes, and when you digest starch, what do you digest it into? Glucose. Yeah, and if you don't use all your glucose, that glucose turns into fat. Your liver does that for you. Thank you, liver. What plant structures store starch? Such an easy question, people don't like it. They're called starch grains. Did you see starch grains under the microscope? Me too. We were making a wet mount of potato that day. What must be added to the potato to see starch grains? Okay, that was all question number 4. If you're following along, that was question number 4. Okay, question number 5. So plants make glucose and then they build it into a structural polysaccharide. Anybody know who that is? It means that it's not for storing energy, it's for building plant parts, and it's made out of glucose. The cell wall is what they build. What's the chemical they build it with?
Cellulose.
Excellent. My favorite polysaccharide, cellulose, is the structural polysaccharide made out of glucose. Can you digest cellulose? No. Naomi already answered this question. What cell part is composed of cellulose?
So wall.
Okay, so eating plants is good for you because they're loaded with cell walls. The crunchier, the better. Whole grains are better than not whole grains. So stay away from white bread, white pasta, white rice, and eat whole grain pasta, whole grain breads, brown rice. It's got more cellulose. Okay, number 6. What term describes the bond between amino acids in a protein? Good. Okay, number 7, I drew it, but just to remind you, we did this activity on page 35 and 36, specifically the table on page 36. So we had water in a test tube, and then we put in some oil. Then we had bile in a test tube, and we put in some oil. And you have to be able to pick up the test tube, give it a little shake, and then figure out which one has the emulsifier known as bile. How are you going to know?
The oil starts to break down.
The oil starts to make little beads, right? Little pellets instead of staying one big clump. That, again, is on page 35 and 36. So that's going to be a skill that you have to show me on a test. Also on a test, one should be able to identify adipose. There's a great picture of adipose on page 37. And then I did an amazing sketch up here. Amazing. What's adipose look like to you?
Your drawing or this one?
The real thing.
Like potatoes.
Okay? The round cells, kind of like the cells of a potato. That's good. Except it doesn't have starch. Adipose cells don't have starch in them. What do they have in them?
Bubbles.
Fat. They're loaded with fat. Okay, still on page 37. I need 3 functions of adipose.
Stores energy, insulates, and protection.
I'm going to use capital E for energy. Is that okay?
Yep.
Insulation. There's no T in there. There. Fixed it. But it's okay on the adipose? Where can you find adipose in your body? Some of us don't want to talk about it. Everywhere. It's under our skin. There's a lot of it under your skin. Right under your skin. And then it's also around your organs. Like your kidneys are going to have some adipose around them to protect them. Behind your eyes, there's adipose, again, for cushioning, protection. Okay, is everybody okay on exercise 3? Chemical composition of cells. Anybody need anything repeated? Okay, cell structure. I'm going to move on back to page 40 again. At the bottom of page 40, we drew on one side some cyanobacteria, and on the other side, human cheek cells. Yes? Okay. And then we answered questions like, are these prokaryotic or eukaryotic? Are they autotrophic or heterotrophic? And so here's question number 1. How do you determine if a cell is prokaryotic or eukaryotic? What do you look for? You look for a nucleus. Okay, what if you don't see a nucleus?
It's prokaryotic.
It's prokaryotic. If there's no nucleus, then it's prokaryotic. How about if it has a nucleus? Eukaryotic. So, guys, I think you figured out these are 2 important words for this class. I mean, you really can't leave this class in December without knowing prokaryotic and eukaryotic. Okay, I think I hit you pretty hard with stuff like this last Thursday. Things cells have in common. Okay, what all cells have in common. I think there's 4 that we've learned so far. Go ahead. Say it again. Oh. I'm back. Thank you, Daniela. I need things that all cells have in common. Not mitochondria. Ribosomes. Thank you. DNA. Go ahead. RNA. Go ahead. Ribosomes, DNA, RNA. The plasma membrane. And that is one of our topics today. Downstairs, we're doing the plasma membrane and how materials move across that membrane. So we're talking about in lecture today. So all cells have a plasma membrane. And this goes back to day 1 together. We went through that list on day 1. It doesn't matter if you're prokaryotic or eukaryotic, you've got all these things. Okay, page 44, I want to remind you parts of an animal cell. You should have done that activity for extra credit. Go back and review. That picture looks familiar. Yeah, that one showed up on the test last week. Some of you were happy about it. Some of you weren't. Okay, page 45, parts of a plant cell. Now, I'm going to go back to page 44. Can you find me on page 44 at least 2 things an animal cell has that a plant cell doesn't? Centrioles. What else? Good. Okay, plants have, animals have centrioles and lysosomes. Plants don't. Let's go to the next page. I need 3 things a plant cell has an animal cell doesn't.
Cell wall.
Good.
Chloroplast, and a central vascular. Wouldn't it be funny if we had a central vascular?
That would be weird. We'd be like,
It'd be like Gumby.
well, or maybe we wouldn't. We would fall over when we dehydrated. Everybody would know if you needed some water.
Getting that dehydrated, you do fall over.
I guess, yeah.
Okay. Oh, let me say one more thing before we go further. All the wet mounts that you and I made that day are on your test. So let me just tell you what wet mounts I'm talking about. The cyanobacteria, page 40, you sketched it. The human cheek cell on page 40. Page 46, the elodia, which is that green aquatic plant. That one's on our test. The onion epidermis is on our test. And the potato is on our test. If we made a wet mount of it, it's on our test. So go back and review your sketches and the parts that you labeled.
So the looks of like the images are going to be on the test or something?
No, I make fresh wet mounts that morning and put them on your test. They are wet mounts. If we made a wet mount of it, it's on your test. If we didn't make a wet mount, then it's not on your test. I'm not putting anything on your test you have not seen.
So page 40 and 46.
Yes, ma'am. Okay, last week, on Thursday of last week, we did some activities on diffusion, osmosis, and active transport. Now, I'm going to delay active transport for a little while because we're going to do that later in the week. But we are going to talk today about how materials move in and out of a cell. And I think you've heard of diffusion before. That's what I'm thinking. In case you don't know diffusion, I found it on page 47. If you will, join me at the top of page 47. Diffusion is the movement of molecules from a higher to a lower concentration. That's diffusion. And diffusion, of course, is due to the constant motion of matter. You know, molecules are moving around all the time. Okay, now I'm on page 47, and I found a sentence that starts with the word diffusion. It says, diffusion is a general phenomenon. Everybody find that sentence? Okay. Well, yeah, diffusion is a general phenomenon in the environment, but I like the next sentence. The speed. Yeah, everybody find that sentence that says the speed. The speed of diffusion is dependent upon such factors as temperature. Now, explain that to me. If I wanted diffusion to go faster, would I heat it up or cool it off?
Heat it up.
Good. So higher temperature will make diffusion go faster because you increase the kinetic energy of the molecules. That's good. Okay, speed of diffusion is dependent upon the size of the molecule. Would small things or large things diffuse faster? Small things. That's good. Okay, then lastly, the speed of diffusion is dependent upon the type of medium. Now let me explain that. If you put a drop of water, no, that's a lie. If you put a drop of dye in a beaker of water, the dye would diffuse through the water, yes?
Yeah.
Okay. If you put a drop of dye in Jell-O, would the dye diffuse? Not right away. Teresa's right. It would eventually, but it's going to take more time because of the density of the medium. Okay, it's easier for the dye to move through water because water's not very dense. It's harder for the dye to move through Jell-O because Jell-O is more dense. And that's what they mean of type of medium. Frankly, the easiest thing to diffuse through is gases like air, and then liquids, and then solids. It's hardest to diffuse through a solid. Everybody shake your head like, well, yeah. But okay on that sentence. It's a good sentence. Okay. Diffusion of water is given a specific name. What's the diffusion of water through a membrane called? Osmosis. That's good. Okay, so I already answered question number 2. In which would diffusion be faster? Dye in agar or dye in water? Now, I crossed out the word agar and put the word Jell-O. Have you ever heard of agar? It's biology Jell-O. It's what we use in biology. You and I are going to use some agar later in the semester, and the day we use it, I'm going to get you to feel it so you'll shake your head. Yeah, it does feel like Jell-O. Yeah. We use agar in biology mostly around in our building to grow microorganisms. But you and I are going to use agar as a semi-dense material to make molecules move through it. So that'll be later in this semester. So we are going to get to use some agar in this class. So I just want to introduce the word agar to you. It's like Jell-O. Okay. What was the answer? In which would diffusion be faster? Water. Okay. And it's because of the density. Good. Okay, take a look at my picture. I drew a picture. First of all, molasses is kind of an old-fashioned kind of material. I don't know if you have any molasses at your house. Have you heard of it?
Yeah.
What is it? It's a real dense, sugary material. It's kind of like a syrup, but even denser. It's a real dense, syrupy material. So do you remember last week we had this tubing and you had to move it between your fingers while it was wet to open it up?
Yeah.
And we twisted it and tied a knot. And we put, starch in the bag. Just pretend instead you put molasses.
Yeah.
Okay? And then again, you twisted the other end. Can you tell from my drawing that's what I'm trying to tell you? Okay. Then you drop this baggie of molasses down in distilled water. And my question is, what's going to happen?
Nothing.
Oh, yeah, something's going to happen. Water's going to do what? Okay, JC, move into the bag or out of the bag? Okay, JC thinks his water's going to move into the bag because water is really high in concentration in the beaker and really low in concentration in the bag. And so water's going to move from high concentration in the beaker to lower concentration in the bag. So what's the bag going to do? Expand. So the bag will swell. Will that make the bag weigh more or weigh less? It's going to weigh more. So you could actually quantify it. You could weigh the bag before the experiment and weigh the bag after. Or you could measure it with a ruler, do its diameter or circumference, something like that. I want to use these 3 words to describe what's happening here. And if you don't remember these 3 words, they're on page 51. Isotonic, hypertonic, or hypotonic. How would you describe the environment that you have put this bag in? Okay, hypo. This was a hypotonic environment. The environment had very, very, very little dissolved substances, and it was real high in water. So JC's right, the bag is going to swell. Thus, it was in a hypotonic environment.
Was it just the iodine that moved in, or was it water and iodine?
I believe water and iodine moved in. Again, we could have measured the mass of the bag ahead of time and then measured it afterwards to figure it out.
That's why I didn't think anything, because I was under the impression it was just the iodine that moved in, because that's what changed the color.
Yeah, I see your point. Okay, I got another drawing up here.
You gotta stick it in
Do
there.
you think the iodine was diluted enough where it could make it into the?
You mean, it depends on the dilution of the starch. If the water content of the bag was equivalent to the water content of the beaker, then it'll be no change in water and just the iodine would have moved. But to know that, we could have measured the bag first and then at the end. Okay, guys, what's going to happen here? Okay, I hear water's going to move out.
The water's gonna stay in, would it not? Because it's less dense on the inside?
Yeah, there's a lot of water inside the bag. Is there a lot of water in the molasses? No. So the water's going to move. It's going to move out. Yeah, that bag is going to shrivel. What kind of environment did I put that bag in? Yeah, that was a super concentrated, sugary environment. It's going to suck the water out of that bag. But okay, on my drawings, they were not in your review sheet. I just threw them in there so we could just talk about it and remember these words. Okay. Okay, so number 4 is considering the function of the contractile vacuole and the protus, which is misspelled. How about that? Protist. I got to fix that. What environment do you predict that an amoeba lives in? Well, I thought you might want a little more information about amoeba, so I did a drawing. And in my drawing, I used the abbreviation N for nucleus. What do you think PM is my abbreviation for? Plasma membrane. An amoeba does not have a cell wall. It just has a plasma membrane. And then CV is contractile vacuole, but some people call it a water vacuole. So let me explain what happens for amoeba. Amoeba fills its contractile vacuole with water until it gets really full, looks like a bubble, and then the bubble moves to the side of the cell and the water is expelled. Then it fills another bubble full of water, then that bubble moves to the side of the cell and it is expelled. Then it does it again, over and over again, to get rid of its extra water. Kind of like how you fill your urinary bladder with urine, and then when it's full, you go and empty it. It's kind of like that. So the contractile vacuole is how the amoeba controls its water. So thinking about that, I'm going to go back to the previous question. What kind of environment do you think an amoeba lives in? And I gave you the 3 choices.
Probably a hypertonic.
So it's shriveling all the time?
Or no.
No. Hypo. It's swelling all the time. What if the contractile vacuole stopped working? What would happen to the amoeba? Yes, it would burst. It would explode. Okay, on number 4. Number 5, explain the results you obtained in the potato strips. Well, that was on page 53. You guys remember these potato strips? We had one that we put in distilled water and one that we put in really salty water. And I did a little drawing up here to remind you. One of these came out really good and hard, rigid. One of these came out kind of bendy. Which one was hard or rigid?
One had no salt,
Yeah.
distilled water.
Kind of environment was that?
Hypo.
Cells swelled. How about the other test tube where the potato came out kind of bendy or limp? I'm sure that's quite a word, bendy. I don't even know. Is that a word?
Yes.
It is today. Bendy. Hypertonic. Good. Which one would a plant rather have? Hypotonic environment or hypertonic environment?
Hypo.
Hypo. Gives it more structure. Plants like having nice, rigid structure. And again, that was on page 53, and we answered questions on number 5 on page 53. We're going to omit number 6, and number 7. But I do want to remind you about a table that we did on page 51. How about that? The blood cell one. Y'all remember the blood cells? That was big fun. I thought that was just tons of fun, looking at those blood cells after you put them in different salt solutions, huh? Yeah, so I want you to know what is isotonic for red blood cells. Right? Look on their table, page 51. What salt concentration is isotonic for red blood cells? 0.9% salt. Can you think of why humans would want to know that?
Because pure water would, make our blood cells explode.
Mm-hmm. Yeah, if you hooked up a patient to distilled water in a hospital, you'd kill them. Naomi laughed. We're talking lawsuits here, people, okay? If you visit someone in the hospital and look at their IV, it's going to say 0.9% NaCl on it.
Saline.
Saline. Mm-hmm.
I did have a question.
Yes, sir.
I thought that would be quite interesting. Is there a way to observe the cells exploding or bursting?
Sure. So what you would do is slowly change the salt concentration. So like you could do 0.8% and they would swell a little bit. 0.7, they'd swell even more. 0.6, so slowly lower the salt concentration until you'd hit the concentration where they would have spontaneously ruptured. Because of time, I just went with 0% salt because I knew that as soon as those cells hit that water, they were gone. Okay, yeah. Okay. Why did test tube number 3, I'm on page 51, why was it on test tube number 3 we could see through the test tube? There were no more blood cells. What happened to them? They burst. So what kind of environment was test tube number 3? Hypotonic. Why did red blood cells burst in a hypotonic environment and potato cells didn't?
Because potato cells have a cell wall.
Potato cells have a cell wall. Okay, that's good. Do that. Don't care about that. So that's answer number 8. You just answered number 8. Okay, I got one more thing to remind you about before we go to pH. And that is the bottom of page 52. We did some sketches. Bottom of page 52. I think they were red onion. Yeah? Okay. I put a red onion cell in a hypotonic environment, and I think it was completely full of the pigment. We shaded the whole cell. And then we took that water off and put salt water on. And then what happened? Shriveled. What kind of environment was the salt water? Hypertonic. So here's why I'm pointing it out. On a lab test, you should be able to pick up a potato and figure out if it was put in a hypertonic or hypotonic environment by trying to bend it. But you can also tell by seeing the plant cells under the microscope. If the cells are completely full, they were in a hypotonic environment. If they shriveled up away from their cell wall, they were in a hypertonic environment. You can tell by touch, you can tell visually with the microscope. But he's okay on that. Great. All right, lastly, pH. Page 54. I got, what's an acid?
Below 7 on the pH scale.
Yeah. It's also a substance that gives away something. Hydrogen ions. Remember that?
Yep.
That's what makes it an acid. That's the chemical property that makes it an acid. And then you guys are right, we developed a scale to show acidity and basicity. All right, so if an acid is a substance that gives off hydrogen ions, then what's a base? Good. I'm going to use the word accepts, takes, that's absorbs, that's all good. Whichever word you like. Okay, so the pH scale goes from 0 to 14. 7's in the middle. What do we call 7?
Neutral.
Below 7?
Acid.
Above 7? And again, a better view of this is page 54. They did a better job than me. If you like their pH scale. Okay, then we started looking at buffers. Buffers. What's a buffer?
Maintains or tries to maintain a neutral pH.
Mm-hmm. Frankly, the buffer that we typically use, you and I will use, is pH 7, but I can buy buffers at pH 9 or pH 5. I mean, there's different chemicals that can buffer at different pHs. All right, I know you like my graph. You drew it too. Which of these graphs, A or B, everybody check out my graph, which of these graphs would be a buffer?
Graph B.
Graph B. Everybody see how it's resisting the pH change. As you're adding acid, it's resisting the pH change. And I think you drew this graph on page 55, correct? Okay. Then lastly, we tested antacids. Everybody remembers Alka-Seltzer. God. Yeah, Alka-Seltzer. And Alka-Seltzer is an antacid. What's the function of an antacid?
It's a buffer designed to neutralize acids.
That's very good. It's a buffer designed to neutralize acid.